Good evening, good afternoon, good morning, depending where you are. My name is Jose Puente, Deputy Senior Editor of the Journal of Medical Microbiology. Welcome everyone to the first session of this year's seminar series of the Journal of Medical Microbiology, and thank you for joining us today. Just a few notes before we start. The live transcript is available if required. If you prefer to hide it, just click on the live transcript button and select Hide Subtitles. The session will be recorded and the link will be sent to you afterward. It will be available to view for 7 days. Please use the Q&A function if you have any questions you would like our speaker to answer during the Q&A time at the end. Feel free to use the chat to introduce yourselves and for general comments. Today, it is my great pleasure to introduce our speaker, Professor Kalai Mathee. Renowned at Florida International University, is celebrated for contributions to microbiology and influential role in academia. As the first FIU faculty inducted into the American Academy of Microbiology, Professor Mathee, a Malaysian native, has achieved notable milestones. Her academic journey spans from the University of Malaya to a PhD in microbiology and immunology from the University of Tennessee, Memphis, and a master's in public health from FIU. Her career features several pioneering roles, including being the inaugural chair at FIU's College of Medicine and the first international editor of the Journal of Medical Microbiology. She became the first international trustee and council member of the Microbiology Society in January 2024. Her mentorship has impacted over 190 individuals, ranging from professors to middle school students, contributing to over 170 publications and 5 patents. Recognitions for her membership include the Faculty Award for Excellence in Mentorship 2011 and the New England Biolab's Passion in Science Award 2014. Professor Mathee has delivered over 150 lectures worldwide and engages actively in FIU's School of International and Public Affairs. She has been instrumental in establishing significant programs like the Global Health Conference and Consortium, and has significantly contributed to the Martin Luther King Forum. Her global commitment to education and science is notable. She was honored with the 2022 Microbiology Society Outreach Prize and has received prestigious awards, including the 2011 President Council World Head Faculty Award and the 21 FIU Faculty Senate Service Award. Mathee's career epitomizes her dedication to the community within FIU and in the broader realms of microbiology and tabular studies. Her resilience, optimism, and perseverance underscore her transformative impact in academia and beyond. Kalai, thank you for being with us today. The floor is yours.
Thank you, Professor Jose Luis Puente. Thank you for the kind introduction. So it's a pleasure to address our audience for 2024. The topic of my discussion today is how do you master a scientific review? And to that, before I start, I have a little disclaimer that, in Tamil, we say, "Katrathu kaiman alavu, kalladhathu ulaga alavu," which simply means that what we know is very little. There's a lot more to learn. That's why I believe in being a lifelong learner. And, sometimes I know a little more than you do, and there are things that you know that I don't. And perhaps we'll teach each other to be more successful. So the presentation outline goes as I'm going to have a very brief moment to talk about what makes a good reviewer. And then to really review a good article is to understand what a manuscript or an article is all about. So we look into the anatomy of an article, and we'll discuss a little bit about the genetic nomenclature. Being a microbiologist and a geneticist, we need to be very cognizant of what is right and wrong. And then we talk about, after you review, what kind of recommendations you can provide. And if we have time, I'll go into back-end process. That what happens when somebody submits a journal, an article, and until to the end of the process where we make a decision to accept or not to. So what makes a good reviewer? A good reviewer provides an honest, objective analysis of the manuscript. The hope is that the editor decides whether the article is worth moving forward or not based on a reviewer's recommendations. It's to provide a complete critique, which is a constructive criticism, but not to criticize. And I do understand the difference between a critique and criticism, right? Critique is evaluate with the knowledge and propriety. Criticism is to find fault in people, right? So that's not what we're doing. We're going to critique a paper. And the other important thing is that if you agree to review, the timeliness of meeting the deadlines. Imagine as an author, you want the reviewer to return the review on time. And same times when you agree to review, you do make sure provide a timely response or inform the editor you need extra time or not. And they can always accommodate whatever your decisions are. And so that will make you a good reviewer. So let's discuss the anatomy of article, and I'm going to go through the various components of the paper, and you know all this if you are already a writer. And let's me go through one by one and talk about what makes a good title and what one should look for. So let's start with the first one, the title, right? The title is the first thing in any anybody who's interested in the science look at. So the title should be convey the reader what is the paper about. You know, it can be concise and descriptive, which is really the core content of the paper. One of my mentors love long titles, and he insists that the longer the better because people will really understand the paper. But it's really, to some extent, it's your choice. You know, like you can have 10 to 12 words, right? And it should never be a complete sentence. And you have to make sure it's not like one single sentence. And the important thing about the title is whether those words that you pick as part of your title can be used as a keywords when somebody is searching for a paper or for a subject matter. Make it catchy, not very pedestrian. And you know, you can be very pedestrian if it's not a scientific article or commentary or something like that. But in a scientific forum, you want to keep your title of your article as scientific as possible. So what do the reviewers do, right? The reviewers is, you know, you're the first thing that you see, right? One of the things I highly recommend is not putting any names of the countries unless absolutely necessary. For example, you are doing a 10-year survey of a particular healthcare practices in a particular country, it makes sense. Otherwise, nobody says, oh, I did this research in UK or US or France or Spain. But whereas a lot of authors will put the name, you know, it's an Iranian study or it's an Indian study or Malaysian study, immediately you will restrict the number of readers who will actually read that article. So don't use the country names unless necessary. Again, no names of hospitals and communities. If you want to talk about it's a what you call a tertiary hospital or secondary hospital, find out the research. It's important people to know the kind of hospital you use. Then you put this whether secondary or tertiary, use a generic name to describe, right? A community, it could be a rural community or urban community rather than putting a name of a community from a particular region. Again, it restricts your readers. Another big important thing is that you don't use any abbreviations on the title. Spell out everything. I never use any abbreviations, including bacterial names, including methodologies. You know, it's like PCR, even though it is accepted, people talk about PCR, it's still polymerase chain reactions. So titles should not have any abbreviation. The other thing is the use of proper nomenclature. And this is probably one of my pet peeves because being a geneticist and trained by an extremely well-known geneticist, I am very particular whether the bacterial species names are properly addressed. Like you never capitalize aeruginosa, for example, right? It's only small c. Even though the Microsoft Word will start correcting it for you because it comes after a period. So it's the second letter is never capitalized. And then you ask the name whether this particular sentence refers to a gene. If it's a gene, it's always italicized. And if it is referred to a protein, the first letter is capitalized and the whole three words or the name of the protein is never italicized. So these are simple genetic nomenclature one should follow when you are writing scientific papers. When I see these, I always find that this person who's writing this paper doesn't really know the genetics nomenclature. It's almost immediately puts me off of the kind of paper I'm going to read. So you got to be very careful. And you can name, if you like the science as a reviewer, you can tell them to correct all this. And you can only do that if you know what to do, right? And also, you should look back at the title after reviewing the paper and ask the question, does it convey to the reader what is this paper is about, right? And is it appropriate? Is it misleading? Is it overstating or understating? Because sometimes the authors tend to overstate and there's not enough information there. And then sometimes there's a lot of information. They overstate when there's no information, and they understate when there's really important finding. And I see that quite often with when I review paper, right? And the question is really, does the title invite the reader to read the abstract? Is that the title impactful? And we ask that question, is the title impactful? In other words, after looking at the title, does the reader goes and says, oh, I need to look at the abstract? So that's what the title should be, right? And recommend changes and make sure the title is of broad interest and it will attract more audience. And the idea is when an author publishes a paper, they want high citation. High citation comes from having good titles. And as a reviewer, you can recommend to tell the authors, please reconsider the titles. You can recommend title exact words, or you can suggest what other things didn't work so that they can think about it, right? Let's briefly talk about bacterial nomenclature. And because this is the microbiology journal that we are talking from, Journal of Medical Microbiology, as you know, most of them are bacterial species, or and you can see that it goes from species to domain. And as you know, when you go to species, genus, and family, all three are italicized. It's always italicized, right? And species names are never capitalized. Remember, genus is italicized, and species like coli is not capitalized. However, several species have zeroas, right? The pathoas and zeroas. When that has happened, then the second word is actually capital letter and non-italicized. A very good example is Salmonella typhi. Salmonella typhi's actual name is Salmonella enterica serotype typhi. In this case, you can shorten it saying at Salmonella typhi, where TYPHI is not italicized and the first letter is capitalized. So always know your species name and find out, if the author made a mistake. Of course, people in the field have been long enough. We know which one has pathoas and zeroas and serotypes. If you don't know, you can always Google and check when you're reviewing a paper. And then often all journals ask for running titles. May not be so important these days with all open access, but however, the running title is short, and normally it's a journal specific. They may ask a number of words. They may restrict you to 50 to 60 characters. It's based on your title, and you create a short version. And there you can actually can use abbreviation. Being catchy is not a priority, but clarity and accuracy is more of a priority, right? And here, like I said, abbreviations are allowed. So for example, here is a long title, regulatory repertoire of Pseudomonas aeruginosa AMP-R, includes multiple virulence genes. So then my running title was P. aeruginosa AMP-R regulates pathogenesis. And it sort of captures what I want the authors to the readers to understand what this article is all about. Then almost all articles ask for keywords. And this is something I don't know where we went wrong. People don't seem to understand the role of keywords. When you do a Google search, right, the Google system will immediately look at the title and abstracts. Then it goes to the keywords. If you use the same words that are already in your title and your abstract, then you're not helping your paper any further. So you want to find who are the others should look at your paper, right? So don't use they should not use any words in terms of the title and abstract. They can look for subject headings in MASH. And I always look at the whole paper and ask my question, do I want the biochemist to look at my paper? Do you want another person from another field to look at my paper? That's when you decide what the keywords are. So not words in terms of title and abstract. Look at the content of the paper, your discussion, and come up with the keywords based on that. So, and then there's often, abbreviations are used within the abstract and within the text, right? And when do people should use abstract? The rule of thumbs is that it appears 5 times. Then you can use an abbreviation. And the count doesn't include the abstract. So you just have to look at the text, how often that comes. And, when they abbreviate, the abbreviation, the first time when the word appears, the whole word appears and the abbreviation is in the bracket. There are people who tend to do the abbreviation followed by the details of what the abbreviation. That's wrong. The right way is to introduce the whole word, put the abbreviation, right? And so very often, of course, the bacterial names are spelled completely when used to first, right? Streptococcus is Aspergillus, and subsequently it's abbreviated. What happens if that particular paper has 2 S, Streptococcus and Staphylococcus? You do not use letter abbreviation at this point, right? And it have to be spelled each time you talk about Streptococcus pyogenes, you got to spell it out. And Staphylococcus aureus is to spell it out. And then don't use 2-letter abbreviation SP. This is often shorthand for doctors. And don't do that. That's not scientific. So abstract should be, often the author should be writing this last, not the first. So it should be concise, complete, simple, and informative. And remember, this is the second thing that look at. So they should persuade the reader. And the number of words or characters varies with journal. And some of them are structured or unstructured. No reference should be there in the abstract, right? And anybody who's not even in the area should be able to read it and understand at least the premises of that paper. So the components of abstract, when you say structured, it has introduction, it has a gap statement, aim, methodology, results, and conclusion, right? We don't have space for discussion here. And even if the journal doesn't ask for structured abstract, the abstract should be created in this order. Then you just remove all the titles. Then it will make a complete abstract. So as a reviewer, you are looking for a very clear and comprehensible abstract. And often what I find is authors don't give enough introduction to their paper. There should be at least 1 or 2 lines that talks about what the paper is all about, and then talk about the hypothesis or the gap statement, then talks about methods, and then results and conclusion, right? So does that abstract have all the components necessary for a reader? And that's what a reviewer should look at, right? And it should invite the reader to explore. And then you also ask the question, is the hypothesis novel? Are they doing something new? Or is it a Me Too paper? Very often, they will repeat the exact same thing in one strain, and then they come back and do the exact same thing in another strain. And that's what I call a Me Too paper. And then if not, then it's good. And then what as a reviewer do, you recommend that, this sounds like a Me Too paper. And often also, you can also find that you can simply do a quick Google to find out it's a Me Too paper. You just type the title and you find it in the Google or a PubMed if this similar paper, or Google Scholar, a similar paper has appeared. And if you have no idea if that's been done before. So let's go to the second part of the manuscript, the introduction. And the introduction really, the role is to introduce the readers to the subject. Remember, not all reviewers are in the field, of the same field. And we often review papers that we are not part of. Even though I'm a Pseudomonas person, I might review another gram-negative or gram-positive. So when I read the introduction, I want to know everything about that particular paper or information needed for me to make informed decision, right? So you want to be comprehensible. It should address whether there's a published literature on that, and whether the literature is critical review, not simply say, this paper states that and that paper states that. No, I don't want that. I want to have a critical commentary on what is the paper all about, right? And then the concept, the idea for the researcher, the article, right? Because when you give a proper introduction, look at the second last paragraph or the last paragraph. Based on all this research, here's the gap statement. This is this paper, what is trying to address. So that is what is your the framing of the hypothesis is all about. That is literally the last paragraph in the introduction. And so the introduction is supposed to provide a balanced view. So you as a reviewer looking at whether the authors have critically reviewed the published literature, have they referenced appropriately? And very often authors tend to do self-referencing. They reference themselves, even though there are papers that predates them, or even some papers that are better references. So you can also check that, right? And you can quickly look at the references and you see how reason is the last reference they used. And that will also tell you something about the ability to reference properly. Right? And then have they, in the introduction, identified the gap or need in the field, right? And then have they framed the concept of the research of the article in their last paragraph? Again, we're going to check whether their hypothesis is novel or is it also a Me Too paper. So these are the things that as a reviewer you're looking at. So that is for introduction. Let's talk about materials and methods, right? Usually this is the easiest to write. Every author should be writing these first. And since it's already something is already done, it should be in the past tense. It includes the design of the experiments and how the data were collected. And often what happens is, even I have been guilty of, I've gone back to an old paper and found out that I forgot to add a method, because we always forget that, when you talk about the results, is every methodology in the results being covered in materials and methods. So it's very easy to cross-reference. So cross-check with the results. And these days, we request that you put what statistical tools were used, because that will tell us whether appropriate statistical tools were used or whether appropriate bioinformatical tools were used to address the hypothesis or the question. And, and every experiment should indicate replicates, whether they are technical replicates, how many samples per experiment were done, or biological replicates is how often the experiments were done. And that has to be indicated in every methodologies they used, and whether they use primary references, because like I told you, they tend to use the self-referencing, or they will give a reference and then you go to that reference and then you'll go to 3 references before you find the primary references, right? And if they say, oh, it was previously referenced, then did they add any modification or they did not, right? And then what kind of units or concentrations we use is very journal specific. You can look at the journal introductions to look at what is recommended ways to present your units and concentrations. Right? So what is the role of an author, reviewer, right? And like I told you, have the authors provided all the methods related to their results. And you can quickly glance at the results and glance at the methods and see if they've done that. And then if are the methods been described as sufficient is someone to reproduce the experiment. And if you read a method and you can't reproduce the experiment, then it's missing details. And people tend to do that, and make sure you check that. And then the other thing that always need to ask is, if they say they use X amount of antibiotics or this kind of chemicals, did they use appropriate amount of antibiotics or chemicals? Is that physiologically relevant? Even when you're listening to a talk in a public forum, you can look at the amount of chemicals they use and says, that's not relevant. Nobody uses 100 grams of something, milligrams, maybe micrograms or nanograms or femtograms. So you want to be very conscientious of how much chemicals they use to get an outcome. Right? So that's what I mean by physiologically relevant. And then, of course, we are looking at whether methods have been appropriately referenced. And then if they have used any human animal subjects, institutional approval and number for their protocol, and if they have any sequences, have they deposited it appropriately. And the other thing is, we talked about appropriate statistical tools. Not all of us are very good at statistical or bioinformatic tools. You can always call upon your friends or colleagues who have a better idea to say, hey, they're doing these experiments. Do you think this is the right statistical tool? Or you can do a search online and say, comparing this, give some keywords and say, is ANOVA is a good method, or is it a t-test a good method? Are they comparing two things? Are they comparing multiple things at the same time? So you really need to understand, and more these days, all scientists are expected to know biostatistics and bioinformatics, and they're getting better at using these tools, but there are still people who don't, and you want to be very vigilant about this kind of analysis. All right, so move on to the next part of the next anatomy of the manuscript is results. So what is the goal of results? Is to present the results as is, unless it's a results and discussion. We can talk about it later. Here we're only considering results. And the results, the idea is to present the purpose of the experiment, how was it done, and of course it should have what were the controls, and it should have outcome of the controls, outcome of the experiment, and results as authors see them positive or negative, right? And you extend, you only interpret and analyze to extend required to transition to the next session. They shouldn't start discussing the results there, because that's what discussion is for. Right? So when you're looking at the results, this is what you're looking for. And if they don't give you the outcome of the controls, how do you trust the results? So it needs to have what was the outcome of the controls first before it give you the results section. So, and the thing is, the question as a reviewer you ask is, are the experiments appropriate to address the problem presented, right? And do the experiments include appropriate controls? And very often they will say something, oh, we use this as a control. And is this a real control? It's just to make up for a line to say it's a control line. No, you want to ask that question. And whether they discuss the outcome of the controls, and again, whether the results are under or over interpreted. And, it can happen to anybody. You can overinterpret because you get overexcited, or you're so conservative, you underinterpret. Right? So you can, as a reviewer, you can suggest what has been done, right? And then if they, again, we talked about materials and methods that are having all the results, methods used in the results, and the other way around. They talk about experiments, but there are no results. And so you want to also check that. And are the experiments failed, right? And are there any experiments that fail that add value to the paper? Maybe they don't talk about it, but then they actually have done it, or they have done it and they don't talk about it. So sometimes negative results add value to the paper, right? That allows them to develop the next part of the paper. So they should talk about that, right? And then you ask the question, what experiments are missing? And I always recommend my students this, right? Don't read the results, don't read the discussion. Just look at the table, understand the table. Look at the figures, understand the figures. Make your own conclusion based on the figures and tables. Then go back and look at the results. Why? Then you know whether they've underinterpreted or overinterpreted, or they missed completely. And then you can really get into the paper and you can really analyze the paper much better. So That would be my recommendation as a reviewer. Just look at the tables and figures and say, okay, this. And that also will tell you how bad the results are, whether even it's worth going to the next step. It's if the result sections have no controls in there, why bother to read rest of the paper? Just base your comments on that, right? So we talked about figures already, and and always choose figures and tables, right? Remember, pictures worth 1,000 words, and you want to try and make it into a figure that's easily visible, visual, and also understandable, and it adds value to the manuscript. And those days there was a journal preference for tables versus figures, etc., etc. This become less important having going online, but I highly recommend figures versus tables. But if sometimes tables are easier to present, right? And the quality of figures, of course, is very important, and that should be these days. Again, it's no money, no huge charges for color printing. So make it color or make it halftone. If it's black and white, make sure you use the right shades. They use the right shades. And then if they use a published figure, make sure in the legend it says that they got the permission to publish this paper figure. And at the same time, as a reviewer, you look at a table and say, oh, this doesn't need a table, right? It can be described in a brief statement in the text. Then you can recommend, this table is unnecessary, should be just added a brief statement. And in the figures, it's not necessary to describe in detail, especially in the legend, but it refers to the methods and materials and method. And if they used abbreviations or icons, make sure it's defined in the legend or in the figure, right? And also results are whether expressed as percentage or absolute values, and what is 100% correspondent must be stated. And again, every results and every table, if it's based on an experiment, it should tell what were the replicates, whether it was a technical or biological replicates, how often they did. And if it's the statistics, what statistical tools they use to get the statistics, right? And usually a figure legend starts with a very short descriptive title and what is being presented, followed by details. And then when they look at the legends, is the legend easy to follow? Sometimes authors are very bad at providing a complete detailed legend, and you need to request as a reviewer for a detailed legend, and whether they are, I need to understand the figure without returning to the text in the results section. So that's the role of legend. Legend has to be comprehensive, and they present it in appropriate format. It's a histogram is a good way to do it, or what kind of plots is a better way to present a particular results, right? And then are there statements of reproducibility? Remember, we talked about that even with tables, right? With the other sections, materials and methods. And again, tables should not be used if can be described, right? And so the difference between a table legend and a figure legend, table legend is always on top of the table, figure legend is always below the figure, right? And it should be short and descriptive, no details. All the details for table comes under as a footnote below the table. So it just should be a short title. And then if you use the appropriate markup symbol, again, that could be very journal specific. They can tell you what markup symbols you used and whether it is what kind of, if it's absolute value, what is corresponding 100%. And again, tables should include statements of reproducibility. Okay. So again, as a reviewer, you are following whether there's appropriate format. Do they have a short description on top? Again, do they include appropriate controls, statements of reproducibility, and is it comprehensible without referencing to the text? Like I told you, table legend, short title on top, and the details of everything you did in the table comes below the table as a footnote. Now move on to discussion, and this is probably the hardest for anyone to write or read. You use an active voice, and how do we write a discussion, right? And how do we create the subheadings? And we talk about how subheadings can be created. And what is the anatomy of discussion, right? Anatomy of discussion includes a first paragraph that sort of restates briefly what we knew, what was done, what was the overall interpretation. That's your first paragraph. Then followed by subsequent paragraphs. And how do we decide what is going to be subsequent paragraphs? Imagine that person gives a talk. That presentation have conclusions, right? Or summary slide. So the summary slide, each summary becomes the title of the subheading, and each or conclusion become a subheading. Then for each, this is repeat, okay. So then followed by a last or concluding paragraph, right? This is the anatomy of discussion. So for each subheading, right, you start with an intro statement. What were you trying to do? And a conclusion, how and you arrived at a conclusion. And how did you arrive? Based on these experiments, we arrived at this conclusion, right? Then the following thing is that how does it agree with published literature? These results are similar to be found as in previously published, which then stops any further discussion. Or if it doesn't agree, it says, oh, these results contradicts previously found in this organism or in this paper, and then ask the question why. Because maybe they used a different strains, they used a different medium, they used a different temperature, right? Everything can change the outcome. So then you have to say why you think they're different, and then you can speculate why your results are different from the published results, right? So for me, when an author speculates, it tells me that the author is really into the research. He is creative, thinking about ways to address the differences, right? And propose what can be done to address the problem. That is anatomy of each subheading, right? And each is the same figure. Okay. Right? And then the authors have to also address the limitations. What are the limitations of the paper or the work they did? Which also tells me that the author is very self-critic about his own or her own research. And it's good to do that. Then you don't have the reviewer telling this person doesn't even don't think of the research retrospectively. So the more you are honest about your limitations, the better the reviewer will look at the paper, right? And you have to finish with the overall conclusion. And as a reviewer, you want to ask the conclusions based on sound data, right? They failed to compare and contrast their findings with other published papers, and if they use the correct references. You can ignore references that disagree with you. And if you have already had a quick search on all the other papers, you have an idea whether they're ignoring or not, right? And address the reasons for difference in the outcome and what experiments are needed to make the appropriate conclusions in case if they didn't complete the experiments. And if they think that they didn't complete, they don't have enough experiments, as a reviewer, you can recommend these may be moved to a different journal like Access Microbiology, which will publish papers that are incomplete, right? As a reviewer, you can recommend that, right? And then the authors can provide details of any future experiments, whether they're going to do it or not. They can say that, this needs all these other experiments that can be done to get to a different conclusion or a better conclusion, right? And if they addressed any gaps in their outcome, their results, right? And then the last thing is the concluding paragraph. And I always look for what is the concluding paragraph? What do they say about their research? And what is the overall conclusion of their paper? And where things need to go next? Again, the concluding paragraph gives a lot of freedom. In fact, discussion gives a lot of freedom to the authors to speculate, to think beyond the immediate research. And we already briefly talked about ethical statements, and they need to make sure it has an approval date and number. And, there are websites for human experiments for UK and US. I'm sure the different countries they have their own websites, and you need to have a statement if you're using human subjects that gave informed consent to the work. And if it's an animal experiment, again, it needs to be approved by ethical committees. Again, the guidelines are country specific. And if you want UK guidelines, there is a UK website, and then the USA guidelines for USA website. And you have to make a statement that experiments do not suffer unnecessarily. And in the approved statement, obviously they have to mention how the animals were euthanized and how much suffering they had to deal with, right? So in the manuscript, they have to say that and make sure that's all stated. And then, so the last thing is the references, right? Whether all ideas have been cited, concept tags, the data that not their own, whether they use primary references instead of secondary references, this like reviews, makes no sense, right? And then the statements and references should match. And often I find, especially with my students, statements and references don't match. So I have to push them to go find the right reference for the right statements, right? And then the other thing is that all references cited in the text must be listed. And this is also could be done at the level of publishing site, but very often, if you don't use a reference manager, or if you don't use EndNote or some kind of reference software, then you will have this problem. If the author uses that, then this problem doesn't exist, because the program will tell you which statements are missing references, or rather which references are missing, even though you have stated there is a particular reference, right? And so that is the anatomy of the manuscript, right? After a reviewer looks at the anatomy, really understands the paper, and the editors often ask you to rate the manuscript, right? The rating of the manuscript, you can rate individual sections and decide what should be the rate, right? And really what goes into deciding whether that manuscript is a high caliber, low caliber, it's a 90%, 80%, or less than 50%, is the subject address is worthy of investigations. Depending on the journals, the caliber of the novelty of the research is very important if you're going to certain journals, right? Is the information presented new, right? And it's incremental. Even if it's incremental, does it add important component to the existing knowledge? And that's what we are looking at. And do the data provided support the conclusions? All that goes into you thinking as a reviewer, there's a scientific content of the manuscript is good. It's the standard of presentation, right? The scientific content may be good, but the presentation is bad, the language is bad, they completely miss the point, and all that go into your decision making. How should I rate this manuscript? And the significance of this paper, because whether it's a new and whether it adds a huge value to the existing literature, the scientific literature in that particular area. And after you do all that, what you need to do as a reviewer is summarize in a paragraph the originality, significance, and acceptability, right? And improvements is necessary. You as a reviewer do not recommend whether you should accept, revise, or reject. This recommendation only goes to the editor, not in the reviewer comments. You can tell the editor, which only the editor sees, that you recommend accept or minor revision, major revision, or reject, not to the authors, right? You are not the decision maker, because the editor will have to look at everybody's review and decide which way to go. And you may recommend reject, the editor might still accept with the major revision because the other editor said accept, and then he or she needs to make that call, right? And again, only to the editor, not the author. And no any other confidential remarks, you can make it to the editor, not to the author. We do as editors review the reviewer comments, and will remove any confidential remarks out of reviewer comments. And that's something editors should do, should review the reviewer's comments and remove any confidential remarks that accidentally were left in the author's comments to the authors. So I think that's pretty much on how to review. And we have used to 45 minutes. I think I still have a little bit time. So I want to go and do the other 2 slides I have before the Q&A. Will that be okay, Dr. Puente?
That's okay, Professor Mathee.
Okay. So anyway, let me acknowledge first. I want to acknowledge the audience for being here, and also Dr. Jose Luis Puente. Professor Puente is the JMM Scientific Seminar Series co-chair. He's also senior deputy senior editor with the Journal of Medical Microbiology under the pathogenesis section, and do reach out to him. And the 2 members of microbiology staff who has been instrumental in helping us today and for the seminar series is Fionn Huff-Watson and Dalia Nicodon, sorry. So I'm going to go to talk about the backhand process. Maybe before I go to backhand process, should I take questions, Jose? Should I continue?
Sure, we can do that, Kalai. Thank you, and thank you for a great talk, and it was very informative and educative. I hope everyone enjoyed. And we have some questions, so let me go through them. So one of the attendees is asking, why do editors of Western journals reject some novel studies from developing countries that doesn't have foreign collaborators? That is, all authors are from Senegal, for example, and they are reporting a novel bacteria. Experience have shown some Western editors reject their manuscript.
My answer to that is, I think it's a very wrong what is the word for it I'm looking for? I don't think that's accurate. I don't think papers are rejected because they don't have Western co-authors. The papers are rejected because there is poor presentation or incomplete presentation, not well articulated arguments. I don't think that's true. And also choose the right journal. If you're introducing finding a new organism, go for IIJSME, there's International Journal for Systemic Investigations. I think, Fionn, can you put the link to that journal? That's one of our society journals that we support. So I think finding the appropriate journals, I think it's the wrong concept you have. I don't think any articles are rejected because they don't have Western co-authors. I would stand by that statement.
Thank you, Kalai. So we also have this question. Where shall we submit if the paper is incomplete? Is it access?
It's a very easy question. So if the paper is incomplete, right, the Microbiology Society has a journal called Access Microbiology. We created the journal because we wanted as a scientist, I know we do experiments and it's incomplete because the student leaves, the project is not going anywhere, but they were good results. And people shouldn't be wasting time repeating those experiments. But there's no place to send them until this journal was created. So in Access Microbiology, you will see articles that are incomplete and hope that somebody who reads it and decides to complete those experiments and come up with amazing outcome. And so I would recommend sending it to Access Microbiology, write in your letter, cover letter, and say that this is the experiment that was done. These are the, hopefully someday somebody will do these experiments and they will review your paper. They will accept the paper if it's written nicely and argued, what I'm missing. And you can go and see that I have submitted article to Access Microbiology. I know that work will never get completed, at least in my lab, right? Somebody else can complete it.
So I will add to this question, what about solid negative results?
Those are also a good place to access microbiology. And you can think about it, well, I would even argue that you can try a regular journal and say that, this is negative results, but we think that it's important for the community to know that this question was asked and addressed, and this was the outcome. And this again, because sometimes the negative results are very critical to move forth. And access microbiology is again another place for you to submit any negative results.
Thank you, Kalai. Nisanart is asking, as many generated AI tools are used in scientific writing, how to check whether paper has been written by AI? Are there any pre-screen from the journal before sending to reviewers?
Well, at the moment, it's a big question, right? If people are using ChatGPT to address, write the articles. Listen, we can't check whether you use ChatGPT or not, but ChatGPT alone cannot create the sections. You have to provide all the information it needs to write the right section. And again, you cannot simply accept whatever ChatGPT tells you. You got to look at the paragraph and correct it and fix it. So I don't have any problem with that. Because if you don't give all the information you have for your article, it's not going to write your paper, sorry. It will write your paper based on the results you provide. And again, if you don't check it, and very likely we'll be able to. Having said that, there are a lot of efforts to figure out whether the entire paper is completely written by ChatGPT. But ChatGPT cannot replace you completely. Without your information, your experiments, your results. ChatGPT cannot write your paper.
And I will add that it perhaps might help with the language usage.
Yeah, absolutely. I agree with you. I even take my whole abstract and run it on ChatGPT and say, have I missed something, right? I can take my abstract and say, clean it up for me. So it takes it and says, okay, here's all the corrections. And I use Grammarly, obviously, right? And even if I use ChatGPT, I take from ChatGPT makes a lot of grammatical errors. Please note. Take that and check it on Grammarly and see if Grammarly finds out if there is a mistake. I'm not proposing that I'm not a Grammarly representative, but thank God for Grammarly. I use it extensively. As you know, English is my fourth language. It's not my first language or second or third. So I have to work very hard to improve my English to where I am right now. Even then, I still cross-check. I listen to what Microsoft Word tells me when I make a mistake. Then I go back and check on Grammarly and make sure I've not made any mistakes. I think everyone should do that.
Thank you. John from Taiwan, thank you for the impressive presentation. Just wondering, does EndNote sometimes go wrong? Do you suggest checking the references style manually?
Okay, thank you for the question and thank you for the compliment. So the EndNote or any reference manager, when you pull it straight from the PubMed or Google Scholar, they do not correct things. So every time you pull a reference, you need to open that reference and make sure they didn't remove the italics of the strain names, bacterial names, gene names, right? And sometimes it will not change the title accordingly. Remember, all the first letters are sentence structure. They're not capitalized depending on the journal. So every time you pull a reference from PubMed, open it and check to make sure the page numbering is correct. If you use EndNote, which I do extensively, I manually make sure my style method says whether the page numbering is 100 to 103 or 100 to 3, right? You can set all that commands in your EndNote. So you can set that up appropriately, but every reference need to be checked every time you pull the reference from the PubMed or from the website, because it will change the name, it will remove the italics, it will remove the italics of the gene names, and it may capitalize, may not capitalize. So you can fix that at that level. And then when you pick the journal format, you can make sure you fix all that. And I would recommend take 4 references, not all of them, and run it in EndNote and make sure it is in the right format. Then it will fix everything.
Bamidel is saying, wonderful lecture regarding the abstract. Some journals specify that the abstract should be around 100 or 150 words. What should authors do in this situation when the abstract should be able to summarize in full the details of the research? So I guess it's meaning that there is not enough space in the abstract.
Thanks, Bamidele. Good to see you there. So Bamidele is our Nigerian champion there, and who has been really promoting our seminar series across Africa, especially Nigeria. So, learning to summarize is an art. You have to, if they say 100 words, figure out what words to drop and still make a complete case. There's no way around it. I think many authors are very wordy, and we need to figure out where to drop things. And you ask the question, if I drop this sentence, will that going to make a difference, right? And these are sometimes these tools, AI tools are quite helpful with that as well. But make sure it doesn't remove everything you want to say, and you can cross-check that, okay?
Will we follow the same rules to review the abstracts as it will be difficult to look at the results? I'm not sure.
I'm not sure the question there. Okay.
Sorry for that. So from the author point of view, what would you suggest if the authors feel the comments from the reviewer are rude?
Okay, so when I talk about manuscript writing, which basically go through the anatomy of article, and I talk about how do you respond to reviewers. Today we didn't have that discussion. Reviewers are not supposed to be rude. Remember, they supposed to provide a constructive criticism, and editors can certainly fix the comments. But most importantly, I think you can tell the editor in your cover mail saying that you thought the reviewer was not, it appears that this reviewer has a conflict of interest, or it appears that this reviewer is very rude, and that editors then will take into account the next time they ask those reviewers. As an author, your goal is to get your damn paper published, right? So you got to be very, very mindful and say, oh, thank you for the lovely comments. Or you can say, we apologize that it was misleading, and so we have rewritten this anyways. You cannot address to the reviewer that you are pissed off at their nasty comments. Those comments, you can send it to the handling editor who's handling your paper, because that will be useful for us. Sometimes editors don't always review the reviewer's comments, and then they can always make a note that this reviews on his reviewer's profile. Okay.
Thank you, Professor Mathee. There's another question here: how to reject the manuscript without hard feelings and still encourage them?
Yeah. So as a reviewer, you have not have to worry. You just tell the editor to reject, right? So you don't have to reject, but you can be nice and say, this paper could certainly be improved, recommend improvement. I like when reviewers give good recommendations, even though the paper is rejected. That allows me to go and improve my paper for the next time when I'm going to publish. So you tell the editor, this paper, whatever reasons you recommend rejection to the editor, not to the authors. But to the authors, you can be very honest and say, this sections needs to be improved. This section needs missing all this, and da-da-da-da-da. And that will allow them to hopefully realize that they need to fix it. And I can tell you, some authors are very stubborn. They won't go and fix even after reviewers told them that they need to fix, they think that reviewers are wrong. If your paper is getting rejected multiple times, you must ask the question, why is it getting rejected? So something needs to be fixed. And you listen to all the comments from all the reviewers and go and fix it. There's no harm done with that.
Samson is asking, in cases where the right equipment or the right resources are not available to do an experiment, but the authors improvise. So what should be the view of the reviewer in case the reviewer feels they didn't do the experiment with the right equipment or the right tools, but they are not available in the place where the work was done?
So that's a serious problem. And, sometimes even in my case, we had an old paper that we submitted and the tools have gone far ahead, right? So our tools we used were old. And the important thing is that you say that in your matrices method, this is within, the availability of that. And in your comments to the editor, when you do response to the reviewers, you talk about the limitations, right? We all know limitations in countries where, not every tool is available. And so you need to describe the limitations and says, in order to address this, how we went about doing this experiment differently to get to the same answer, and explain the methodology that was revised in order to get there. I don't think we're going to penalize because you don't have a next generation sequencing tool to do something, right? And so I think it's all, for me, it's that be honest, be clear, be open, and you're good. And if you think that, this is not going to cut it, send it to Access Microbiology. Within the limitations of the study environment, this is what we were done.
From Kalpana, how can you identify a conflict of interest as a reviewer?
A conflict of interest as a reviewer. Yes, first of all, you know the author very well. Maybe you know the author and you don't like that person, or maybe you like that person, you don't want to reject. That's a conflict of interest. And for me, I know the author and I don't respect the author. I immediately say, I don't want to review the paper. I don't have to explain to them why. You just say there's a conflict of interest. If you have published with this person, it's a conflict of interest. If you are, we recommend that if you're in the same institution, same department, all that things can contribute to be a conflict of interest. When in doubt, ask the editor. I know this person, blah, blah, blah. I think it's a conflict of interest, but I'm unsure. And the editor can always tell you that's your conflict of interest. When I see some articles, when I ask to review an article and I know the authors, I tend not to review too much. If Dr. Puente asked me to review his paper, I probably won't review it, even though I don't publish with him, because I don't want to feel bad about saying something or making a decision. That becomes conflict of interest.
Thank you, Professor. From the author side, I have encountered several reviewers trying to have you cite several of their papers, even if they are not within the scope of the manuscript. What will be your recommendation how to handle those reviewers' comments?
Very simple, right? In the reviewer's comments and says, this is an interesting recommendations. However, this particular paper is not part of the scope, so we are not including this reference. Look at the references and say which one works and which one doesn't work. If it doesn't work, just state it. It is true, and this is why I say making proper referencing from the author is very important. And because you don't know when the reviewer who's reviewing is in that area, oh, he or she didn't reference my paper, right? And that's like a negative, immediately negative feeling that the reviewer has. So referencing appropriately is very important. And if it's a recommended reference is inappropriate, say it nicely in the reviewer comments. this is an interesting paper. However, I don't think it has it's not part of the scope of this paper. I don't think they're going to kill you for that, or they're going to reject your paper because of that. And very often, remember, unless the reviewer's comments were very, very drastic, the paper doesn't go for second review. And so the editor has to make a call based on your response to reviewers. If you don't respond appropriately, then it goes back to the same reviewer, who then can be even more harsh. So make sure you address every comments in line by line, item by item, and address everything. And then as an editor, I'm going to look at the reviewer response to reviewers and say, okay, address, and color code it. Make sure the editor can go and look at your Word document to see where the changes have been made.
How can we identify plagiarism if we don't have the right tools to do it?
Yes. So that's a big problem, right? Grammarly definitely has the option for plagiarism. You can just copy the abstract and do plagiarism. Or sometimes I'll just copy the entire abstract and put it on a Google Scholar, and it might find me articles that are very similar. And sometimes I use my own tools that given to my institution. We are trying to make sure we can check that. And I do believe, I can't remember exactly right, at some point JMM does a plagiarism check, and some publishers do have a plagiarism check integrated into their offerings. And so, it's very funny. And there was recently a paper I sort of felt it was plagiarized, and I went and did a search, and then as you review many papers, you get the feel for it. I've seen it here or something, and then obviously it was a plagiarized. It's the same results elsewhere, and we had to call on the authors.
Thank you. From Samson, how can we access a waiver as students who need their work to be published?
Oh, right. So, there is, if you go to microbiology website, and Fionn maybe can find that, there are countries that fall into the category of, I can't remember the name of list of countries. There are a list of countries where publishing charges are waived. And you should check if your country is one of them, and then you just remind the website, the editors who are reaching out to you. You're publishing from this country, it's supposed to be waived. You can also write to the editors, who then can also check back and see if there's anything can be any compensation may be made. This paper is fantastic results, and we should publish it and come up with the potential ways to publishing it, right? So there are ways and means. You reach out to the journal or the journal handlers, the editors, and the staff can certainly also find out if there are any other ways to get your article published. And I would highly recommend, there's a publish and read model where the university pays a single fee, and then everybody in that university can publish free if the university buys this single fee payment, right? And if your university, there are a lot of researchers who are likely to publish in Microbiology Society Journal, you can send me an email and we'll connect you with the right people to see if they can talk to our staff members. I think Alex Chan is our staff member who pursues that, and he can talk to your university and maybe convince them to buy that system to allow you to publish free. Yeah.
Some of our attendees would like to know how they can contribute to the seminar series.
At the moment, thank you for asking the question. If you're interested, do send us, but I think we have for 2024, we probably still have all the editors presenting. We did have non-editors present at this thing. And if you're interested, and do reach out to us. And at this moment, the committee is made up of me and Dr. Pente. We can review your request, and we can figure out how to include you as one of the authors. As you know, our JMM seminar series have a huge international footprint, over 80 countries, and this is a great forum to present your work. All right. Jose, this is about 10:10. Can I finish the last two and then take back the question?
We appreciate. We'll try to answer some more questions through the chat, and yeah, please go ahead, carry with them.
And we may have a little bit more time after that, because I really want to present this. So one of the things that people don't realize is that there's a lot of work goes behind the scene. And one of them is what happens once you submit a manuscript, right? And there is a pre-quality control check for the manuscript, whether they have all the right things. And once the pre-quality control check, that is done by the staff, which then they assign an editor based on your topics, your title and your keywords. Hopefully they will assign you the appropriate editor, and the editor has an option to say yes or no, whether it's a conflict of interest, or the editor has no time, or whatever. Not, the editor can reject the assignment. But once the editor says he or she will accept the assignment, they will then assign a reviewer, right? They are supposed to get at least minimum of 2 reviews. And sometimes we ask and ask and ask, and we don't get any reviewers consenting. That poses a very big problem. I recommend that the editors tell the authors that we cannot find a reviewer, and we potentially may have to pre-screen reject so that they can go to another journal. Once the reviewer assigned and the reviewer sends the comments, and I request you be timely about it, and then it goes to the editor assigned, and then editor looks at the reviewer comments, and he or she can pre-screen reject, after the pre-quality control, editor can pre-screen reject. Once he decides to go through the assignment, looking at the reviewer comments, the reviewer again can reject, right? Based on your comments, or based on the comments, the editor says, you have to do a revise. It's a minor or major revise. And depending on major, it usually goes to the assigned reviewer. If the reviewer agreed to re-review, if not, he or she has to find a new reviewer. And then the reviewer comments comes back. And again, editor makes a decision based on the reviewer's comments, based on your response to reviewers. The assumption is, don't assume it will be accepted. Even at this point, your paper can be rejected because you have not addressed the comments. And then if the editor says, okay, I will accept, then it goes to the publishing department. The next time you hear from the journal is you get a Gali proof. Gali proof is the document where you are asked to read and find all the mistakes added during the typesetting or any corrections made by copy editors. You'll ask to correct. Excuse me. And so that's what happens. Excuse me. I do want to talk about the Journal of Medical Microbiology scope. We cover clinical microbiology, AMR, both resistance and countermeasures. We look at microbial diseases, pathogenesis, etc. We looked at public health microbiology, which includes epidemiology and any kind of evolution ecology. We do cover veterinary microbiology, especially those are associated with also human infections, so basically zoonotic infections, and One Health. And it covers everything, not just bacteria, viruses, fungi, and parasites. And we're talking about infectious. If it's not the infectious organisms, we certainly have Journal of Microbiology, the journal called Microbiology. And we also have a section called JMM Profiles. That's if you want to talk about a particular organism by itself, it's very prescripted. There is a template that you have to follow. You can do that. I do want to consider attending this Pseudomonas conference, which is in 1st to 5th September in Copenhagen. And with that, I end my session. Give me 1 minute, Azib, before you ask next question.
Mhm.
Sorry about that.
Yes, okay, thank you.
Mm.
We have a couple of additional questions. Our attendees would like to know about why are the impact factors important?
Okay. All right. Okay. You know, the impact factor, history of impact factor is very interesting. Was created by a library to see how many readers are reading it. Even though all journals, many journals said, don't care about impact factor, most people still look at the impact factor before looking at the quality of present publications. I think it's only important to some extent. What's important is getting your science out. If it's a good science, it doesn't matter where you publish. And with PubMed and Google Scholar, all articles show up, whether you publish in Nature, Science, or Journal of Medical Microbiology. And if it's a good paper, it's going to get selected, it's going to be get read by. The emphasis on impact factor is certainly overemphasized. And then there were journals that were formed, the very high impact journals. I'm talking about high impact, not impact factor journals. Even though they do not want any impact factor, these industries keep giving us impact factor on those journals. So I think pay attention to what you write, what you publish. Important is to publish. And I think, and then sometimes you got to look at what the journal publish and how easy to publish. All that does matter, okay? Thank you.
And perhaps I will add, Professor Mathee, that especially for microbiology society journals, they are non-profit. So authors should also consider that by publishing in society journals, they're contributing to the development of the society. So as you were saying, their papers are going to be advertised in PubMed. So the important thing is to publish and people to see the research. So consider society journals as an option without worrying for the impact factor.
Thank you. Thank you, Jose, for the comment. It's so important because Society Journal supports the grants, your travel grants, your conference grants, attending the conference, all that because the societies publish, and this is what our major source of income, right? So do publish with society journals, and society journals are often free, right? And until they go open access, they might cost money. And again, there are ways to counter that. And I would recommend society journals, any society journals, of course. We want you to publish in Microbiology Society journals.
I guess that there is another question. Gopal would like to know, how can new students start writing thesis, articles, and abstracts?
Repeat that again.
So Gopal would like to know, what will be your advice for new students to start writing their thesis, their papers, or their abstracts for meetings?
So I would say, writing a paper is like baking a cake. You follow a recipe, you get a cake, or you get a flopped cake, right? So for me, like what I discussed today, every section of the paper is like a little recipe. Follow the recipe. Start with the materials and method first. That's the easiest. Then go write the results. Then you can say, okay, these are all the things I covered in the results. How much introduction should I give in order to get to the results? Then once you do that, you make your subheadings, do your discussion. Again, I gave you the recipe, how to write a discussion. After you've done all that, ask the question, what does my paper talk about? And there is a very set ways to develop the title. You start from a very big title to lower to the smallest denomination to get to what you want to say in the title without losing all its meaning. Only then you go write your abstract. Abstract should be the last one to write. Again, follow the recipe, gap introduction, gap statement, materials and methods, results, conclusion. So for me, writing a scientific paper is nothing but a recipe that you got to follow very carefully to make it work.
Thank you. I think that's all the questions we have. Oh, just give me 1 second. What's your suggestion for a cover letter? Such as the length, the contents. Your silence, Professor Mathee, your mic.
I apologize. So it's a very easy, right? The cover letter is obviously, make sure you address the editor, all right? Dear editor, blah. And then you say, this is the first paragraph is, this is the title of my article written by blah, blah. And then the first paragraph basically second paragraph rather, you should say, why is your paper important? How is it different? And what value does it add to the literature, right? And then you also want to say that, we have compared and there's no other publications related to this, or there are publications in other organisms, but our finding clearly states that, in Streptococcus mitis is very different from Pseudomonas aeruginosa. Sometimes, you have the same gene in different organisms, they work completely different. So you can say that, this gene was found in this organism. We also found the same gene. However, the whole mechanism of action is different. What it impacts is very different. And then you always end with the paragraph saying that it's not been submitted elsewhere for publications. And then we hope that this is acceptable for the journal. That's, I think, basically, again, another recipe, right?
Thank you, Professor Mathee. So to our audience, please check all the information that is being provided, the webinar chat about the society, about the journals. You're going to find very useful links there. And probably we can wrap up now. Just to have a minute to let you know, well, first of all, to thank you, Professor Mathee, for a great talk. I think it was very useful for everyone. And just let me tell you that the link to the recording will be emailed to you and will be available to view for 7 days. And the next session of the Journal of Medical Microbiology seminar series will take place on Friday, February 23rd from 14 to 15:30 hours UK time, when Professor Peter Coyle, who is affiliated to the Hamad Medical Corporation in Doha, Qatar, and the Biomedical Research Center of the Qatar University Health, and the Wellcome Wilson Institute of Experimental Medicine of Queen's University in Belfast, United Kingdom, will be delivering a talk titled "The Role of National Influenza Centers in Preparing for Seasonal and Pandemic Respiratory Viruses." So we hope to see you during the next seminar series. And once again, thank you very much, Professor Mathee, for your talk, and to our audience for being with us today.
Thank you for hosting us today, and thank you all. Have a great weekend.